Hey everyone, welcome. So, first of all, there comes the codominance. What is codominance? In case of codominance, both the alleles are expressed independently. As a heterozygote, they are expressed independently and equally, and they both represent the homozygous parents. Now, one example is the ABO blood grouping system found in humans. This ABO blood grouping system is mainly controlled by three alleles, namely IA, IB, and small I. Now, IA and IB are dominant, and small I is recessive. Here, blood groups are the phenotypes, where the genotypes are the IIB combinations. So for the blood group or for the phenotype A, blood group A, we have IA, which is the homozygous dominant, and IA small I, which is the homozygous recessive. It forms the blood group A. Now for blood group B, it is there IB, which is again the homozygous dominant. It is because IB is a dominant allele. IB, and in another case for blood group B, we have IBI. Here IB is dominant and small I is a recessive allele. So it is a heterozygous dominant. So blood group B is formed in this way, and at last we have the O blood group O, which is the small I, is the homozygous recessive type. And we now see the combination done by Gregor Johann Mendel, which is the combination between the pure tall and pure dwarf plants. For pure tall, we have the capital T plus capital small t from 2n gametes are present. Alleles are present 2n. When the gametes are formed, then the 2n becomes n. I mean it actually divides into half, and here comes n. So capital T comes from one side, which is from the pure tall side, and small t comes from another side, which is the pure dwarf side. They merge with each other, forms a fusion, and this process is called the fertilization, and as a result a zygote is formed, which consists of the allele formation of capital T and small t. Now this is called the F1 generation. I have written that. Now here comes the F1 generation. Now when self fertilization is done between a member of capital T small t and another member of capital T small t, this process is called fertilization because the genotype is completely same. Then the outcomes we get is capital T, capital T small t, capital T small t, and at last we get small t, and this we can get using the Punnett square, 4 by 4, 2 by 2. And here the phenotypic ratio will be 3:1, 3 tall and 1 homozygous recessive, which is dwarf. It is a pure dwarf plant, pea plant, and the scientific name of pea is Pisum sativum. Underline, binomial nomenclature said by Carolus Linnaeus. You see the spelling right there. Now for the genotype ratio, we have 1:2:1. It is because 1 represents capital T, and 2 represent capital T small t. There are 2 capital T small t, and at last the 1 we have, it represents small t, which is the homozygous recessive. So capital T, it is homozygous dominant. There are 2 capital T small t outcomes, which are the heterozygous dominant, and at last we have the small t, which is the homozygous recessive. Now all we know, it is a star point, that heterozygous recessive is impossible. It is because when a dominant and a recessive allele is present in a certain gene, then we always know it is a factor, it is a result due to complete dominance. Always from a heterozygous pair of a dominant allele and a recessive allele, only the dominant allele will be able to express itself due to its dominance, and the recessive allele will be suppressed due to its recessiveness, and you will just get suppressed and repressed, and it will not get expressed as a result. Now we have an example, we have a case which is called the monohybrid cross in incomplete dominance. Again I say the case name is monohybrid cross in incomplete dominance. Monohybrid cross in incomplete dominance. Monohybrid cross means there are two plants, and in between the two plants only one contrasting character is present, and when cross is done between them it is called monohybrid cross. Due to mono means one, so there is only one contrasting character present. And incomplete dominance means the dominancy by the dominant allele is incomplete, not 100%, it is not complete. So the example we have here is dog flower, also known as snapdragon, also known as Antirrhinum sp, underline as per binomial nomenclature by Carolus Linnaeus. So for the true red we have capital R, and the true, and for the true white flower we have smaller. So here capital R is coming as a gamete, and here smaller is coming as a gamete and forming capital R smaller allele, which is known as F1 generation again. And if we do the self pollination between the F1 generation of two members, then we get as a result capital R, smaller. We can get four outcomes from the Punnett square of the self fertilization of two members of F1 generation. Now the capital R, it means the homozygous dominant we get completely red, and at last we are getting the smaller which is completely pure white flowers we get. But in between there are two outcomes which are the capital R and smaller, and another capital R smaller. There are two outcomes in between capital R and smaller. Now here, due to incomplete dominance of the monohybrid cross, we get a pink color which is an intermediate between the capital R red and white, which is the intermediate between the capital R red and smaller white, and thus we get, as a result we get a color which is pink. And that is why the genotypic ratio is as it is 1:2:1, and also here exceptionally the phenotypic ratio is also 1:2:1. So for in this case the phenotypic ratio and the genotypic ratio is same, 1:2:1. Now, what are the reasons for Gregor Johann Mendel's success of his experiment? The first reason is to consider one character at a time. He considered one character only at a single time. Second reason is his methodological approach. He approached everything methodologically. And the thirdly, he used statistics, mathematical statistics to scientifically, in his scientific experimentation, which was not accepted in his time when he did this experiment, and that is why sadly he was rejected by many scientists, by many famous scientists, renowned scientists in his prime. And he also used clear-cut variables. Next, coming the law of segregation, which is also known as the first law. Firstly, it denotes no mixing of gametes. The gametes will not mix with each other. Secondly, it also denotes the allelic pairs separate during the gamete formation and randomly unite at fertilization. When gametes are formed, the alleles separate or segregate. That is why it is, for the word separate, it is known as segregate. To separation and segregation have the same meaning, and that is why this law is known as the law of segregation. At the time of the gamete formation, the allelic pairs separate, and at the time of fertilization they just randomly unite, but there is no mixing of the alleles, no mixing of the gametes, no mixing of the alleles, there is no mixing. This law was law of segregation, also known as the first law of inheritance, maybe. Yes, first law of inheritance. Then comes what was the time of Mendel's hybridization? It was from 1856 to 1863. How many years is that? '63 and 56. 63 minus 56, how much? 7 years. Yes, almost 7 years. 1856. 18, was it 1856 to 1863? Yes. And in his experiment, he considered seven different characters, seven contrasting characters um, in the plant P, whose scientific name is Pisum sativum. Again, binomial nomenclature as said by Carolus Linnaeus. The spelling of Carolus Linnaeus is quite hard. Okay, and the binomial nomenclature also you have to underline the Pisum sativum part. Now I said the years 1856 to 1863, 7 years almost. And then so there are 7 pairs of characters. So if I have to calculate how many plants were considered in the experiment, so 7 into 2 equals 14. 14 plants, 14 pea plants were considered in the experiment done by Gregor Johannes Mendel for his hybridization experiment of hybridization. Also the hybridization spelling is also hard. All total 14 plants were chosen by Gregor Johannes Mendel in his hybridization experiment. The 7 pairs of characters are as follows. First, we have the plant height, 1 gone. More 6 are present. Flower color, flower position. Plant size is already done. Flower color, flower position. Seed color, seed shape. Pod color, pod shape. For the plant height is axial and terminal. So axial shows capital A and terminal shows small a. And for flower color, we see the pure red and white. Red is the dominant capital R and white is small r. Now red or violet is formed. Red or violet, which is dominant and is denoted by the capital R notation. Now flower position. Okay, flower position. Yeah, flower position, axial and terminal. Axial is capital A and terminal is small a. Flower color, flower position, plant height finished, and then comes 3 finished. More 4 are present. Flower done. Now seed color and seed shape. Seed color is capital Y, small I, Y, yellow and green. Seed shape, inflated and constricted. No, I think rounded or wrinkled. Capital R, smaller. Yes. Then pod color, pod shape. Capital G, small g, green and yellow. Pod shape, inflated, constricted. Capital I, small I. Yes, done. 7 pairs of characters were done. Now at last we have that a sentence. The genes of these 7 pairs of these characters studied by Gregor Johannes Mendel are located on 4 different chromosomes. Like these 7 pairs of characters which were studied by Mendel are now present on 4 different chromosomes.